Morning, everyone. My name is Mafalda Isidro, and today I'm presenting my thesis work that was done on Galway Bay, Mayo Institute of Technology, and my work was about the effects of non-mechanical harvesting of Ascophyllum nodosum on Vertebrata lanosa and its importance for biodiversity. Ascophyllum nodosum is a brown alga of the order Focales that forms extensive beds in the intertidal rocky shores of the North Atlantic. It creates a dense canopy, which forms a complex habitat for other species, such as algae, invertebrates, and fish during the tidal cycle, acting as a foundation species by regulating the community composition and richness, as well as the keystone species, because it controls the biophysical processes with an effect extending far beyond the foundation of the habitat. The biotic assemblage associated with Ascophyllum includes Vertebrata lanosa, an obligate red epiphytic algae that grows after thalli. Not much is known about their relationship, if it is commensalistic or parasitic. However, Vertebrata sustains an invertebrate community that relies on it for food and habitat. Harvesting of Ascophyllum has been an important activity in the west of Ireland for a very long time, and it has economic importance for the local community. There are two types of non-mechanical harvesting used in the area. The first one is hand harvest, that uses knives to cut the Ascophyllum, and the second one is boat harvest, that uses cutter rakes, as we can see in this image, the cutter rake, to cut the seaweed from a boat. But possible effects that harvesting Ascophyllum may have on the growth and survival of Vertebrata and the macroinvertebrate community is not very well known. Therefore, assessing environmental impacts and habitat resilience is crucial to ensure the sustainability of the seaweed extraction industry. The overall goal of this study was to establish if harvesting Ascophyllum affected Vertebrata lanosa and is associated by biodiversity. The study aimed to evaluate the presence and biomass of Vertebrata lanosa and diversity and abundance of the associated fauna before and after the harvest, using two non-mechanical harvesting methods, boat and hand, the one that I just mentioned, and studying if there was a difference between two types of underlying sediment, hard and soft sediment. And at the end, this work may help to develop a more sustainable harvest and resource management methods. The two hypotheses used for this study were, first, harvesting has a negative effect on Vertebrata lanosa biomass and associated fauna, and second, hand harvest is, in theory, more detrimental due to the higher extraction rate, which I'm going to explain why a little bit later. The samples were collected in Kinvara Bay, which is located in the southeast of Galway Bay. A baseline survey was conducted in July of 2020 before a harvesting event in September. A month after, in October, a post-harvest survey was conducted to study potential effects that harvesting had on Ascophyllum nodosum habitat. Here you can see a drone footage of the area showing the location of the three plots that were set up for the experimental harvesting. The first one, hand harvest, as I said before, uses knives to cut the seaweed. And here, almost all the biomass is removed, being the extraction rate around 80%. In the boat harvest plot, it uses cutter rakes from a boat to cut the Ascophyllum. In here, only the topmost part of the thalli is cut, being the extraction rate around 30%. In here, so only the topmost part is cut, leaving about 15 to 20 centimeters of standing thalli still attached. And the third plot is a control plot that was left untouched. In here, you can see a scheme of the experimental design. So in the baseline, 3 replicates per type of sediment were randomly collected using a 50 per 50 centimeter quadrant. In September, during the harvesting event, all the plot was harvested, which is a big square. According to the extraction rates that I mentioned for boat harvest and hand harvest plot, and the control that was left untouched. In the post-harvest, the same was done as in the baseline. 3 replicates per type of sediment were randomly collected for the 3 treatments. And all the material within the quadrant was removed, as we can observe here, before and after. And all the biomass within the quadrant was, the weight was measured on field, and when arrived at the lab, it was frozen until the first stage of sorting, where branches of Ascophyllum that contained Vertebrata were separated and stored in IMS until further sorting. And the second stage of sorting consisted of the separation of the epiphyte and the fauna associated. The fauna was enumerated and identified as much as possible to species level, and Vertebrata was dried at 60 degrees for 24 hours and then weighed. To test hypothesis of whether there was an impact of harvesting from the experimental harvesting method and sediment type, different analyses were performed. For the Ascophyllum nodosum and Vertebrata lanosa biomass, a three-way ANOVA was performed, where time, method, and sediment were the different factors used. For the fauna associated, univariate and multivariate analyses were performed. For the univariate, diversity indices such as abundance, number of taxa, Shannon-Wiener index, Margalef's index, and pilus eveness were calculated, and a ANOVA was also performed to see if there were some significant differences. And the factors used were time, method, sediment, and Vertebrata lanosa biomass. In multivariate analysis, a K-dominance plot, a Simprov test, a Simpro analysis, an MDS, a BV step, and a ANOVA were performed. The results show that there was an overall significant decrease in Ascophyllum nodosa biomass after the harvest. This decrease was of 38.3%, and this may have consequences for the epibiote associated. The hand harvest plot observed a higher decrease in Ascophyllum nodosa biomass compared to the boat harvest. However, no significant difference between the two types of harvesting methods was detected. The hand harvest plot observed a higher decrease in biomass compared to the boat harvest. Let me repeat. The hand harvest plot was the only one that observed a significant decrease in biomass before and after the harvest. Before is represented in green and after is represented in red. And this observation can be related to the habitat conditions, where this plot was considerably more bouldery. It can also be related to the randomness of the sampling, where a spot without much biomass was selected, or even to spatial variability. However, it is still important to say that the boat plot still observed a considerable decrease, but showed not to be significant. There was also a significant difference between the two types of sediment after the harvest. This is because hard sediments, rocky substrates, are more suitable habitat for the Ascophyllum to attach. Therefore, there is a higher biomass in the hard sediment samples than in the soft sediment samples. It was also observed a significant decrease in Vertebrata lanosa biomass of 57.9%, and this may have negative impacts for the invertebrate community. It was observed a decrease across all the three plots after the harvest, meaning that the control also observed a decrease in biomass. And I remind you that the control did not suffer a harvesting event in September. And these results can be explained by the phenology and natural variability, can also be related to the randomness of the sampling, or even to human error. Once again, no significant difference between the two types of harvesting methods was detected, which means no significant difference between hand and boat, and no significant difference between the two types of underlying sediment was detected. This is because there's a similar trend of less Vertebrata after the harvest, indicating that the removal of Ascophyllum led to a significant decline in Vertebrata in both types of sediment. Now, moving to the fauna associated with Vertebrata lanosa, the univariate analysis showed an overall decrease in biodiversity after the harvest. The results confirm that the total number of taxa, Shannon-Wiener, and Margalef's index experienced a significant decrease after the harvest compared to the baseline. As you can see here, the red box plots, which correspond to the post-harvest, are most of the times lower in the graph than the baseline ones. And this means that the community is less diverse and simpler. The factor method was only significant for abundance. This is because the control has quite a different pattern compared to the other two. By this I mean, in the boat and in the hand plot, we see that there is a decrease, more in the boat than in the hand, but in the abundance, in the control, the abundance is quite similar. The factor sediment was only significant for Shannon-Wiener. Although the baseline values of Shannon were quite similar, the post-harvest ones were relatively different due to the increase in data dispersion in hard sediments, as we can observe here, in here, and also due to the big decrease in the control, as we can observe here and here. And this can be explained by the small-scale variability found in benthic communities. PLO's evenness was not significant in any of the factors. This can be associated to a decrease in abundance across all species, but especially those that were a lot more abundant in the baseline, increasing the assemblage evenness, even if it's less diverse. Vertebrate lanosa biomass recorded across the experiment was a significant predictive factor for abundance, total taxa, and Mergelev's index, indicating that Vertebrata lanosa can function as a keystone species. I could conclude this because, for example, if you look at this graph, total taxa, we see that Vertebrata lanosa biomass, when it's low, it has a big impact on the number of taxa that is present. But as it increases, it has less significance. Moving on to the multivariate analysis, the K-dominance plot show a clear decrease in diversity and evenness after the harvest for the two types of harvesting methods and control, as well as for the two types of underlying sediment. One can conclude this because there's elevation of the curves after the harvest, which are indicated by the color red. The Simprov groupings coincide with time of sampling, regardless of method, suggesting that changes in the community might not be solely related to the experimental harvesting, but also influenced by seasonal changes. For example, if you look at the group D, which is represented by the blue square, we see that all of them belong to the same survey, the post-harvest survey, but they belong to different treatments. The same for the group F. They belong to the same survey, but to different treatments. The BVSEP results and Simpro analysis are complementary to each other. The taxa found to be responsible for the dissimilarities between groups are most of the times the same taxa as the ones obtained from the BVSEP. This includes isopods, different species of amphipods, gastropods, and insect larvae. All these organisms have a role in the ecosystem. They play a role in the food web, being a food source for other species, such as birds, fish, and large crustacea. Therefore, if there is a decrease in biodiversity and a change in species composition, it is going to impact the whole community associated with the Ascophyllum nodoso habitats. The NMDS plot for time method shows a clear shift in the community after the harvest for all the three plots, meaning that the control also observe a change in the community. We can observe here that the baseline samples are mostly grouped here, and after the harvest there is a dispersion of the data. And because there was a change in all the three plots, we can say that there were some temporal differences. Now in the NMDS for time and sediment, there is some separation in the baseline between hard and soft sediments. However, after the harvest, there's a dispersion of the data. It appears that soft sediments did not observe a major change in species composition apart from some samples, while samples from the hard sediment look like there is a large dispersion, meaning that there is some change in species composition and structure. Decrease in Vertebrata lanosa biomass seems to be responsible for the groupings. How we can read this graph is, so the bigger the circle, the higher the biomass. So the samples with high Vertebrata lanosa biomass are grouped at the center of the graph, corresponding mainly to the baseline survey, which is indicated by the letter B, while samples with low biomass are dispersed on the graph, corresponding mainly to the post-harvest survey, which is indicated by the letter P. And these can indicate that there is an effect on community composition and structure according to Vertebrata lanosa biomass. However, the Pomanova test showed it to not be significant, meaning that there might be other factors that are driving the community. To conclude and to make some final remarks, I would like to say that Vertebrata lanosa decreased in biomass. However, it is not possible to conclude that these changes were solely by the activity due to changes observed in the control plot, which can be explained by seasonal and spatial variability. Loss in biodiversity and changes in the community were also influenced by cumulative effects of seasonal fluctuations and spatial variations. The high heterogeneity of benthic habitats may have obscured the detection of canopy harvesting events in the present study. Due to the marine benthic assemblages being highly sensitive to variations in time and space, making it difficult to analyze effects of impacts. Some future considerations and suggestions is to include another site in the study. This would increase the cover to a wider range of Vertebrata lanosa spatial variability. This is actually already putting into place in the project where my thesis is inserted in, and also perform the baseline survey closer to the harvesting event. This would reduce the seasonal effects on the community. Thank you so much.